Today we are discussing about structure of DNA. So we will discuss a general structure of the DNA. Then later on we will discuss the Watson and Crick model of DNA, which is a B type of a DNA. Let's have the structure of DNA. What DNA consists of? DNA consists of, or it is made up of, it consists of polynucleotide chain. So it is made up of many nucleotides. So these nucleotides are linked to each other by the help of a phosphodiester bond. We'll see later on how two nucleotides join together to form a phosphodiester bond. And a long chain of nucleotides, we can call it as a DNA. Or we can also call it as a polymer of nucleotides. You can use the word polymer of nucleotides. Now, which monomer units are present in case of a DNA? Which type of nucleotides are present in case of a DNA? We call those one as a monomer units. The monomer units which are present in case of DNA is a deoxyadenylate. Deoxyadenylate. That means deoxy here represents the sugar. Deoxy here represents the sugar, deoxyribose sugar, and adenylate is representing the adenosine triphosphate. Similarly, we have deoxy gammanylate. That means it represents deoxyribose sugar and the guanosine triphosphate. Okay. Similarly, we have deoxycytidylate. That means it represents that we have a deoxyribonucleic sugar and cytidylate is actually the cytosine triphosphate. Similarly, we have deoxythymidylate. That means it represents that deoxy is actually the sugar, deoxyribose, and thymidylate is actually the thymidine triphosphate. So some of the authors, because this is only present in case this type of nitrogenous base or this type of nucleotide is present only in case of a DNA. So some of the authors, they represent it with a TTP, that is thymidine triphosphate. Thymidine triphosphate. The reason is it is not present in case of the RNA, so there is no need to represent a deoxythymidylate here, because we know it is present in case of a DNA only, that means the sugar will be always deoxy here. So no need to represent with a deoxythymidylate, but some authors use a simple name for it, that is a thymidine triphosphate, that is TTP. Now we have the most important thing, that is nucleotides. If we see the nucleotides are attached to each other. Nucleotides are attached to each other by a 3-5' phosphodiester bond. This is the most important thing. That is the nucleotides in case of a DNA or in case of a RNA, they are attached with the help of a bond that is known as a phosphodiester bond. We will see how this bond formation occurs in case of a DNA, and we will also check up why this bond is known as a phosphodiester bond. Like take the two nucleotides. Say for instance, if I am taking here one nucleotide in general, say this is a structure of a pentose sugar. See here at carbon number 1, we have attachment of adenine, so it becomes a nucleoside adenosine. Okay, so this will be position 2, 3, this will be 4, and position 5. We have here a presence of phosphate. Especially if the nucleotides have to join to each other. If the nucleotides have to form a phosphodiester bond, if they have to join to each other, they should be in a triphosphate state. You have to keep in mind the nucleotide should be in a triphosphate state. And one more thing, that is, if another nucleotide has to come to bind the nucleotide, it binds onto the bottom of this nucleotide. So we can say the next thing is, next nucleotide binds on bottom. ये bottom पे इसके जो है bind करता है. So it binds onto the bottom. Now the most important thing which you have to keep in mind, that is why the phosphate. If we have a 5 carbon here, if we have only the one phosphate here, see if I am representing it with the actual structure. Say we have CH2, then we have single bond O, single bond P, then double bond O, then single bond O dash. And here we have also the O dash. So if it is only the one phosphate, then we can call it adenosine monophosphate. But it have released the 2 phosphates as a pyrophosphates. Pyrophosphates. Because it has to now make the bond with another nucleotide. If the next nucleotide comes here, if I am representing with the red color, see if we have next nucleotide, it comes out of the bottom, but most important thing is it should be in a triphosphate state. Say we have here at carbon number 1, we have thymine bind here. So we can say it is a thymidine triphosphate here, and this is the carbon number 2, this is the 3, this is the 4, and this is the 5 here. But you have to keep in mind the most interesting point here we have H, OH, and here we have also OH, H, and here we have H. Now listen to me what happened in next one. Here we have also H here, OH has been 1. Here we have also H. Similarly, we have here H, OH at carbon number 2. And carbon number 3 we have OH, H here. And similarly we have a carbon number 4, H at 5 here. Now listen to me, we have the bond formation that is the phosphodiester bond. Now this is in a triphosphate state. The nucleotide which joins with the next nucleotide is in a triphosphate state. So we can say the 2 phosphates are released here as a pyrophosphate. Because the 2 phosphates are released by this thymidine triphosphate as pyrophosphates. They are released as pyrophosphates. Now what remains here is only the 1 phosphate, and we show the representation of that phosphate. So here we have CH2, okay, located. Then we have the phosphate here. Now this OH group it has lost, it combines with the phosphate group of this one gets released as a water. It is released as a water. Now what happened here? Now we have P here, and we have CH2O here. Then it makes we have a P here and double bond O dash here. And here also we have a single bond O dash here. And at carbon number 3, we have a carbon number 3 with the oxygen, and making the bond with that. Now this is the bond form which actually takes place here. So we have CH2 here, then it's binding with the O because we have O here, then we have P double bond O, P double bond O, then we have O negative by this another O, it makes a bond with another nucleotide that is it. So this carbon here we have 5 and this is 3 carbon. We call it 3 dash, 5 dash, and the bond we call as the phosphodiester bond. The reason is, if we take an ester here, ester we have R single bond O, single bond C, double bond O, and then after it will be anything, it will be R dash. Now this is an ester, but here we have R single bond O, single bond P. Instead of carbon we have P here, then we have a double bond O, then we will be anything R, it will be R dash. Now we have to make 2 diesters here. Now how it gets formed? If you take this is the carbon, making a single bond, so we can say R here is represented by CH2, then making a single bond with oxygen, then making single bond with P, then single bond, double bond with O. So if we take this line, this is a 1 ester bond formation here. It's a 1 ester bond. And if we start from this carbon number 3, this is also a carbon. Now carbon with oxygen, then with the phosphorus, then again with a double bond, this one, like this. We make this one more ester bond. So that is why we call here the formation of diester bond. So instead of carbon, here we have a phosphorus. We have to keep in mind. If we will take a next nucleotide here, so as to understand more how this phosphodiester bond is formed, see we have another nucleotide here, which I am making roughly. So this is another nucleotide, and this will be this carbon number 1. See here is a cytosine attached to it, and here is the H. And we have here OH, we have here H and OH, H and H here on carbon number 2. We have H and H here. Sorry to make the correction here. We have H and H, H and H. Similarly, we have H and H. And here we have OH and H. And then here we have H and carbon number 5 we have CH2. Now, it also comes in a CTT state, triphosphate state, that is cytidine triphosphate. What happens? It releases two phosphates as a pyrophosphates. Two phosphates are released here as a pyrophosphate. The one phosphate remains here. Now it makes post-step. This is the single bond with oxygen, then with the phosphorus. Then here we have a double bond with it. It is a double bond. Then after we have here a single bond with dash. Now what is released here? Now what remains here in the carbon? So we have the single bond oxygen, then we have the single bond phosphorus, then double bond oxygen. Now this is actually known as, this bond is known as here, is a phospho diester bond. So instead of here, if we take how the phosphodiester bond is formed, start from this is a carbon attached to the oxygen, then attached to phosphorus, and we have double bond oxygen. This is a one phosphodiester bond. Here we see this is CH2, okay, attached to the oxygen, then again with the phosphorus and double bond this thing. Now it becomes a phosphodiester bond. So we simply say the nucleotides in a DNA or even though in a RNA, they are linked together by a phosphodiester bond, which is from 3' to 5', because here we have a 3' and this is a carbon number 3, and here we have a 5' which is a carbon number 5 of the another nucleotide. So the bond is being formed between them. We call this bond as a phosphodiester bond. Now one of the important things which we have to discuss about this is about the Chargaff's rule. We have an element Chargaff, one of the scientists, who says, we take an element Chargaff. This element Chargaff states that after studying the DNA of many species, he said that the adenine, that is our purine, in a DNA is equal to thymine. So we have element Chargaff. After studying DNA of various species, after studying DNA of various species, he observed that in case of DNA, adenine, which is a purine, is always equal to thymine. That means how much of the adenine is present in a DNA is equal to the thymine, and how much of guanine is present in a DNA is equal to the cytosine. So we can say this is actually the equal concentration. That means adenine in a DNA, adenine is equal to thymine in a DNA, as well as guanine is equal to cytosine in a DNA. So after observing the DNA of the various species, we find out that the purines are equal to adenine and guanosine are equal to pyrimidines. That is equal to the cytosine and thymine. This was observed after studying the various DNAs of the various species by the Edwin Chargaff, and he gives this conclusion that the adenine is equal to thymine as well as guanine is equal to cytosine. Similarly, he says the purines are equal to the pyrimidine. But it was not realized at first by the people, by the scientific. It was not realized. It was not earlier realized, not realized by scientific world. Scientific world has not realized or accepted this thing. But later on, discovery of double-stranded DNA. Later on, discovery of double-stranded DNA has made this concept acceptable. Now, after the discovery of the double-stranded DNA, what happened? This concept which was given by the Edwin Chargaff, it was accepted. Because in a double-stranded DNA, we have adenine equal to the thymine. The reason is how much concentration of adenine we have in a one strand, because adenine always binds, have a hydrogen bonds with thymine. So that means how much adenine we have on one strand, same number of thymine will be on another strand. After that it was accepted. But you have to keep in mind, the Chargaff's rule is not accepted by, is not applicable for single-stranded DNA and RNA. So it's applicable only for the double-stranded DNA or double-stranded RNA. We can also say it is accepting it, because in a double-stranded DNA we have adenine on one strand and thymine on another strand. It makes a two hydrogen bonds. Similarly, we have guanine on one strand, cytosine on another strand, and makes a three hydrogen bonds. That means how many number of adenine are present on one strand, similar number of thymines will be present on another strand. Similarly, on one strand, how many number of guanines will be present, that will be equal to the cytosine of the another strand. This was given by the Urban Chargaff, and we will study this rule in detail when we will go through the Watson's and Crick's model. So tomorrow we will discuss about the Watson's and Crick's model of DNA, which is actually the B DNA.